this is a joint presentation, as you can see on the front, on the first slide, is from my laboratory in Göttingen, as well as here in the faculty of the University of Buenos Aires, where I have what's called the Laboratorio Max Planck de Dinámica Celular. And I'm very happy that the Max Planck has agreed to let me conduct this laboratory. Usually when a director of the Max Planck retires, they amputate his hands and feet so that he doesn't ask for money. But in this particular case, they were very kind, and I must admit I've been in the Max Planck Society now for 45 years, and they've supported me very well. It's the only job I've ever had. So I'm very happy to be in Germany, but also to have the opportunity to be, of course, in Buenos Aires, where I was born. Now, the presentation also from the lab of Ellie Harris-Erichmann, whom we've heard about because she was a group partner of the Max Planck. She was a collaborator of mine, and she contributed a lot of the ideas and a lot of the workers who worked on these projects. And we've been supported by what's called the Toxic Proteins Project of the Max Planck Society. It's interesting that institutes can get together in the Max Planck and then can do things together. They get the money from the president if he agrees. And this is an excellent means of conducting difficult work in difficult areas. And there are also other units in the government that have supported us both there and, of course, the laboratory of Eli Harris here in Buenos Aires. Now, this picture I showed before, the so-called Grupo Harris, that is continuing the work of Eli, and these are some of her students and postdocs, and I must say they're working really well, and I'm very happy that, especially that Lia Pietrasanta and Fernando Stefani are taking on the job of helping these people meet their needs and do good work. Now, this is a picture of Argentina. We're here in Buenos Aires, and this is the rest of the country, as undoubtedly some of you know. But the real picture, the real map of Argentina looks more like this. And there you see Buenos Aires. There's a little bit of Córdoba, so I don't know if Pincho Cáceres is still here, but he'll be happy to see that Córdoba is there. I'm sorry, Luis, but Mendoza has disappeared somewhere. Tucumán is there. But, and of course, the president of the country, it's good that she's not here because Santa Cruz is barely in evidence. So there is an emphasis, of course, on Buenos Aires, and this leads me to show you this slide because we have collaborations in La Plata and Santiago del Estero and Tucumán, so I feel that I should get a medal from the CONICET or whatever for being a good citizen of the whole country. These are our collaborators elsewhere in the world outside of Buenos Aires and Göttingen. And here are what I call the neurodegenerationists, not degenerates. And again, they're people from Argentina and Germany contributing to all this work. Some of them are from the Harris group, others from this Max Planck lab at the UBA and La Plata. So this chap is from La Plata. So these three people are postdocs, and this is my wife and colleague, Donna, who is probably the best confocal microscopist in Germany, and she has trained many people from Argentina, including names on this list that have been through our lab. Now, I'm going to tell you 4 O. Henry stories. Now, O. Henry was this gentleman over here, shown here, has a real name, but he was known as O. Henry. He was an American writer in the last century who wrote stories with twist endings. Borges did some of the same. And these are great stories, even now, and I'm sure if you go down to the Feria de los Libros in the Rural, you can probably get this book of his stories. But I call them 4 O. Henry stories because the endings were also rather unexpected. And I'm going to start then with this one, the story about intermediates and the aggregation of alpha-synuclein, what we call the acunas. I'll tell you what that is. Then membrane interactions of synuclein and A-beta, the amyloid proteins involved in Parkinson's and Alzheimer's disease, mitochondrial interactions, that's where the action is in these diseases, and stem cell research, which is devoted to also the same topic. Now, amyloid, you all know, are protein structures, these long, elongated structures in which proteins, many proteins, almost all proteins can acquire if they can assume the secondary structure characterized by cross-beta sheets. So this seems to be a universal principle of proteins as polymers, and it's interesting that the end results are always pretty much the same. Now, they cause diseases, and here is a list of some of them, not all of them, and you'll see in red those attributed to the protein office in synuclein, so that's why they're called synucleinopathies. And of course, the most important of those, or not the most important, depending on the person who has them, is Parkinson's disease that affects so many people in the population. Now, these diseases differ in the proteins, the distributions in the nervous system, the cell types, and whether they have intracellular or extracellular deposits. So their deposits have this fibrillar component that I just showed you, the amyloid structures. And they appear in different, they're located in different parts of the nervous system, for example, in the brain. So you have Alzheimer's here, and Parkinson's here, and so forth. So this is something to understand why different cell regions, different regions and different cells are affected in these diseases that have so much in common. Now, Parkinson's disease, a little to tell those of you who are not informed about this, infects 1 or 2% of the population over the age of 65. Some get it young. Clinical signs are movement disorders, as you all know. You certainly have seen people with Parkinson's. The diagnosis is not available. It's just a clinical picture. Therapy, there's no cure. One can use palliative administrations of drugs, replacement drugs, and maybe even gene transfer stem cells, but there is nothing available at the moment. I hope Christian Giesinger, the next speaker, Gaby Fernandez later on, maybe Walter Stummer as well, will give you some insights into what's available at this time. The pathology is simply loss of neurons in the dopaminergic pathways that have to do with movement. And these typically in Parkinson's show inclusions in the cytoplasm. These are called Lewy bodies, things over here. And they are located in the substantia nigra in the brain, in the midbrain, as shown in this image. Now, the genetics is complex. There are 15 genes. One of them is, of course, alpha-synuclein, of which they are familial mutants indicated here. Parkin and pink are involved in mitophagy. They are important in this disease as well. And there are other proteins listed here. The one of interest, most interest perhaps, is LRRK2, because this is a strange, very complex molecule with unknown function, really. And this particular mutation shown over here is the most common genetic cause of Parkinson's disease. The interesting note is that almost all of these proteins have to do with mitochondrial function and their interactions, physiological and pathological, and almost all of them dimerize. This is an interesting feature that has not been really dealt with in the literature very much. Now, we have to use many techniques to study such a situation. They're listed over here, and in red are those that are new here in Buenos Aires in the labs that I've mentioned, for example, fluorescence microscopy of super resolution, and I'm very happy to indicate that this is being employed for these studies now as well. And in addition, the Max Planck Society made available to the group here a microtiter plate reader, which is a very advanced technology. It's probably the only machine of its kind in Argentina, and it's going to be invaluable in these studies as well. It's being used a lot, and it's available to anybody interested in using it at the university in Pavillon 2. Well, the Bermuda triangle of Parkinson's disease is indicated here. This ties mitochondrial dysfunction with oxidative modification, protein degradation. This is where the action is, most people think, in these diseases. And it's the loss of the energy producing machinery of the cell, the mitochondria, of course, that leads to neuronal death. The neuron is a very sensitive cell. It's at a level of activity that's very high. It has to support these long processes such as axons and provide them with the nutrients and with the components, and of course that leading to the synaptic mechanisms that ultimately are required for signaling. And so anything that interferes with that is a problem. And listening to the train here led me to the following consideration. I, of course, like many of you, have been reading about this train accident, this tragic train accident a couple of months ago in which so many people died and got injured. Now, the train was traveling only 20 kilometers an hour when it came to the station there at Once, and yet all this happened. And why is that? I mean, what happened really is that there was a loss of space, and so people got crushed, and there were new structures formed. And this is because of what's called momentum. It's the mass times the velocity that's important in a case like that. And I'd like to think that one can perhaps use this concept of momentum in a biological sense, talking about biological momentum in this system, because if things pile up in the cell, then you generate new structures, and you saturate systems that are present for doing the normal housekeeping of a cell. Most proteins synthesized in the cell are degraded right away because they're not good enough. And if the system then gets slowed down, like when you get older, like in my case, my mitochondria are working half as well as the younger people in the audience, as those are the younger people in the audience, then the cell can suffer. And neurons are particularly exposed to this, and especially dopaminergic neurons, because oxidative modifications are common in the dopamine pathways. So I can't go into details here, there really isn't any time, but to emphasize that environmental, genetic, and endogenous factors are really responsible for pathology in these systems. Well, synuclein is a small protein, 140 amino acids. It's mainly in the brain. It's unfolded. It's one of these strange unfolded proteins. Function not known. Here are some ideas shown. One really doesn't know what it's good for, and we'd be better off without synuclein probably in our bodies. It has this kind of a structure, an elongated structure as shown here, a N-terminus, a core region that is responsible for formation of the amyloid structure I mentioned before. In the C-terminus, it's quite acidic and in fact hinders aggregation, as a student, Carlos Bertoncini, working in Christian Griesinger's department, was able to show some years ago. Well, what about the aggregation of this protein? Well, it's thought that this unfolded form forms some kind of oligomeric species, rich in beta sheets, and they go on to form the amyloid fibril. You'll notice all of these question marks. Almost nobody knows anything about, we know almost nothing about this mechanism. And probably each one of these question marks, if you could do something about them, would earn you a round-trip ticket to Stockholm. So this is why so many people work on it, not because of Nobel Prize, but because this is an important biomedical problem to solve. So we have to understand especially what's happening here, because it's thought that the oligomeric species are the most toxic ones in this system. The factors influencing aggregation are many. They're listed over here. The list could be extended a lot. I've done it this way. I mean, one can think of many ways to influence the formation of structure by a protein that's unstructured at the outset. Polymerization is a little mysterious. You start off in vitro with a protein solution, you expose it to a higher temperature, nothing happens for a while, and then all of a sudden it takes off and you form these amyloid fibrils. So the big question is, and that's why it's shown over here, is what's happening over here? And the reason that we don't know much about it, is that there are few species there. The intermediates are low concentration. Also, the probes we've had up to now have not been very effective. Well, that's why we spent quite a bit of time in this area developing new probes, and this is one of them, the tetracysteine tag. It's the only good expression probe, in my estimation, for synuclein. It's the work of Julia Roberti, who got her degree here with Ellie a few years ago, and she just published her 6th paper based upon her thesis work. And she used a lot this so-called flash compounds. These are biarsenicals. This is a fluorescein thing. You won't be, I'm sure you don't see it on the screen, but this is a fluorescein with 2 arsenate groups, and they combine to a tetracysteine sequence shown over here, 2 cysteines, pro-glyve, 2 cysteines, and she added this as a fusion peptide to the C-terminus of synuclein. And this works really well, and one was able to do work in vitro and in cells, living cells, and the last paper had to do with super resolution microscopy applied to this system. Now, the most recent development here in Buenos Aires at the UBA by these people shown over here is the development of bipartite biosynthetical tags, that is half cysteine or 2 cysteines in a molecule, with the hope that this can give insight into interactions between molecules, for example, the formation of dimers. And this, in a way, complements the extensive use of monocysteine mutants in multiple positions in the sequence, alanine to cysteine transitions. And these can be applied in polarity and position-sensitive probes for looking at many processes, and I'll come back to this. And there have been non-covalent probes as well in the past, and including quantum dots that you heard about nanoparticles just now from Pedro, and nanogold. And Soledad is from Córdoba, and she worked on the amino naphthalene sulfonates and non-covalent ESIP probes. I'll tell you what ESIP is in a minute. And the most recent thing has been the use of fluorescent amino acid analogs to make synthetic A-beta peptides. This is the Alzheimer peptide. This is the work of Schwarczak in our lab, postdoc, and I'll show you some results with that. Well, these are the chaps who made the ESIP probe. This is an ESIP molecule. I'll tell you how that works. Note that it has a hydroxyl position in this part of the molecule. And Dima Yushchenko was a chap I sent here to Argentina, and Jonathan, who I see is sitting in the audience, is a graduate student here, was a graduate student of LA, and will finish this year. You see that these people are very happy, not because they work with me, probably because they work with Ellie, but because they were able to synthesize this molecule, and they could label them as synuclein at these 3 positions, and many others in the meantime, and do things with them. And this paper was published in 2010. Well, this is how an ESIP molecule works. You have a ground state, and when this molecule gets excited, then you have the potential for a proton transfer from this position to this position. And if that happens, then you generate what's called a Tautomeric species. And the nice thing about that is that the original so-called N species and the T species have different emission bands. So you get a dual emission, the M band, the T band. And this is very useful because as a function of polarity and environment, you get very big changes in the spectra, intensity and the ratio of these 2 bands. And so these are ideal probes. In my estimation, they're the best probes for studying protein-protein interaction, protein-membrane interactions at the moment. Now this was the idea, label synuclein with this stuff at the cysteine positions and expose it to conditions that lead to aggregation, a little bit of the protein label, not too much. And then hope that you see intermediates with different properties than the mature fibrils. And that's in fact what we did, what we saw. Tremendous changes in the T band, the Tautomeric band, as the molecule aggregated and became less polar. And if you look in more detail at this, you can see that every conceivable fluorescence property of this molecule changed in a way that was interesting and revealing. And in fact, the normal assay with amyloid detecting probes such as thioflavin T goes something like this in this aggregation reaction. So it takes this lag time and maybe 20, 30 hours to see something, but these probes were doing very well at this point. All right, so then Luciana Giordano came to our lab for a while, and she and Jonathan have been synthesizing many other compounds, and they have been very successful in this, and the newest gold standard for sulfatochromosm has just been published in this journal. Well, now let me get to the acuna family, synuclein amyloids. This is from a paper that Jonathan Feuerbach, who's really not only a brilliant chemist, but has golden hands for AFM studies. We published this paper, and we said that we had evidence for intermediates that were non-canonical in the sense of not being amyloid, not having beta structure, but that and were soluble and diffusible, and they probably might be involved in toxicity. And this started many years ago, if you like, with alpha-synuclein and AFM. We showed that on the microgrid you can see the formation of intermediates, but in solution this was a different story. Now these are what amyloid fibrils look like at the end, and what Jonathan was able to see were intermediates that are